Welcome to the Smoke on Pharmaceutical and Medicinal Chemistry. Module 1 of this course is Introduction to Pharmaceutical Chemistry. The third part of the module is Chapter 1.3, which is Metabolites and Antimetabolites. The instructor for this chapter would be Dr. P. Bhushanavati, Controller of Examinations and Selection Grade Lecturer in Chemistry, St. Joseph's College for Women Autonomous, Visakhapatnam. So far, in this module, we have learnt about terminology, drugs, receptors, mechanism of drug receptor interaction, types of receptors, pharmacodynamics, pharmacokinetics. We have learnt about agonists, antagonists in pharmacodynamics. We have also learnt about absorption, digestion, metabolism, excretion in pharmacokinetics. We have also understood the difference between the two terms, and now we progress to chapter 1.3 on metabolites and antimetabolites, which are the molecules present in living systems. The expected learning outcomes from this chapter are: the learner would be able to identify and classify the compounds in living systems as metabolites and antimetabolites and also recognize their importance, distinguish between metabolites and antimetabolites based on their function, comprehend some important functions and applications of both, and also figure out the risk in applying antimetabolites for the treatment of cancer. Contents of this chapter are: Introduction to Metabolites, Classification of Metabolites, Functions and Applications of Metabolites, Antimetabolites, Applications of Antimetabolites, and a word of caution on antimetabolite uses. Introduction to Metabolites. Metabolites are small organic molecules that are involved in various biochemical processes within living systems. They are the intermediates, the products, and substrates of metabolic reactions that occur within cells to sustain life and maintain various physiological functions. Metabolites play a crucial role in energy production, growth, and regulation of various cellular activities. Examples for metabolites are antibiotics and pigments. Classification of metabolites. Metabolites are classified into primary and secondary metabolites based on their function. Primary and secondary metabolites are the two categories of molecules produced by living organisms, but each serves a distinct role in their growth, development, and interaction with the environment. Primary metabolites are organic compounds that are directly involved in normal growth, development, and reproduction. They are carbohydrates, lipids, amino acids, nucleic acids, and sterols. They are considered essential for all living organisms and are found in plants as well. Secondary metabolites. Secondary metabolites are also called natural compounds or phytochemicals. Secondary metabolites are molecules that are not directly involved in the core metabolic processes required for growth and development. Instead, they often play specialized roles in interaction with the environment, in the defense mechanism, and also in the communication system. Secondary metabolites are usually produced in response to a specific stimulus, such as a change in environmental condition or some stressor or during some signaling pathway. Primary metabolites are carbohydrates, lipids, nucleic acids, proteins, etc. They are very much essential for basic life processes. Their production and regulation are closely linked to the metabolic state and growth of an organism. They are found in all living organisms, that is, plants, animals, and human beings. Secondary metabolites, often uniquely found in specific taxonomic groups. Secondary metabolites are specifically found in groups, in specific groups of plants and animals. They are more or less unique to the particular species. They have secondary biological function. They are the pigments, the fragrance agents, or the defense mechanism. Their useful applications include dyes, medicines, drugs, and toxins. But the function of many secondary metabolites is unknown even today. Examples for secondary metabolites are alkaloids, flavonoids, terpenoids, etc. You can see here organic compounds that do not participate directly in the growth and development of a living organism are called secondary metabolites. Some examples are shown here. You get alpha pinene from the pine tree. We get the piperine from red and black pepper. These are some examples. There's the rose flower. From the rose flower, you get the cyanidin, which is the coloring pigment. So here we have shown the compounds as well as their sources. Cactus plants, you get the mescaline. Functions of metabolites. Metabolites are further classified based on their functions. Energy metabolites. These metabolites are involved in energy production as well as storage. Examples include ATP, which is the primary energy currency of cells. ATP stands for adenosine triphosphate. And also nicotinamide adenine dinucleotide, which is NAD, a molecule that carries or transports electrons during energy producing redox reactions. Then you have the building block metabolites. These are the small molecules that serve as the building blocks for the synthesis of larger molecules. Amino acids, which are the building blocks of proteins, and nucleotides, which make up DNA, are examples for building block metabolites. There are the signaling metabolites. These metabolites act as signaling molecules that communicate between cells and regulate various cellular processes. Examples include CAMP and CGMP. CAMP is cyclic adenosine monophosphate, whereas adenosine triphosphate is the energy currency in cells. Adenosine monophosphate is the signaling metabolite, and cyclic guanosine monophosphate, which are involved in intracellular signaling pathways. Waste product metabolites are also generated in living systems due to metabolic reactions. For instance, carbon dioxide is a waste product of cellular respiration, and urea is the waste metabolite generated due to protein metabolism. Secondary metabolites also play important roles in defense, communication, and interaction with the environment. Applications of metabolites in medicine. Metabolites find application in disease diagnosis and they are also used as biomarkers. Changes in metabolite levels can be indicative of various diseases and metabolic disorders. By comparing metabolite profiles between healthy individuals and patients, doctors can gain insights into disease progression and develop early diagnostic tools. So, by monitoring the changes in metabolite concentration, diseases can be diagnosed. Pharmacokinetic and drug monitoring. Many drugs undergo metabolic transformations in the body, resulting in the formation of metabolites. Monitoring the levels of these metabolites helps to determine the effectiveness and safety of a drug. It also aids in designing appropriate dosage regimens for patients. So, drug monitoring and pharmacokinetics can also be followed through metabolite concentration measurement because drugs are metabolized into secondary products. If we can monitor the concentration of these metabolites, you will be able to understand the drug pathways better. Drug development. Metabolite analysis is essential in the process of drug discovery and development. They help to identify potential drug interactions and adverse reactions that may arise due to the interaction between a drug and its metabolite. Personalized medicine. Metabolite profiling can provide insights into an individual's unique metabolic characteristics. That is what is meant by personalized medicine. Metabolite profiling can provide insights into an individual's unique metabolic characteristics. This information can be used to tailor medical treatments or drugs to an individual's personalized needs, which is known as personalized medicine. So, metabolic profiling can be different from one person to another. If you do the metabolite profiling, we can design drugs better. We can design medication personalized to a particular patient. So the basic applications in medical field of metabolites are in disease diagnosis and as biomarkers, for pharmacokinetic studies, for drug development, as well as personalized medicine. To continue with their applications in medicine, they are also used in monitoring and treatment progress. Monitoring changes in metabolite levels during the course of treatment can provide valuable information about the effectiveness of therapies. How effective the particular treatment is can be arrived at, or you can identify the effectiveness of a particular therapy by monitoring changes in metabolite levels. Nutritional assessment. Metabolite analysis can be used to evaluate an individual's nutritional status and dietary habits, which are indirectly indicating the food from which they are derived or the nutrient from which they are derived. So, this concentration is indicative of the original concentration of the nutrient or the food. Toxicology and environmental exposure. Metabolite changes can indicate exposure to environmental toxins or pollutants, helping to identify potential health risks and design interventions to minimize their impact. So, environmental exposure to specific pollutants can trigger the production of specific metabolites in living systems. So, if we can identify those metabolites and identify the source, then the intervention strategies can be implemented. Understanding disease mechanism. Metabolites are also helpful in understanding disease mechanisms. Metabolite analysis contributes to understanding the underlying mechanism of disease, how the disease is triggered, how the disease progresses, what is the current status of the disease. All such information can be obtained through metabolite analysis. It provides insights into how metabolic pathways are altered in various conditions, paving the way for the development of targeted therapies. Just as personalized medicine, you can have disease specific therapy if we can do metabolite analysis and we can follow the progress of the disease. Antimetabolites. Antimetabolites are another class of drugs that interfere with the normal metabolic processes of cells, particularly those involved in DNA and RNA synthesis. These drugs, there's one small clarification here. Metabolites are natural chemicals. Antimetabolites are drugs. So they are a class of drugs that interfere with the normal metabolic processes of cells, particularly those involved in DNA and RNA synthesis. These drugs are designed to mimic the structure of a natural metabolite that cells need for growth and division. However, once incorporated, antimetabolites hinder or block essential cellular processes, leading to the suppression of cell growth, division, and proliferation. So this kind of treatment or property of antimetabolite is used in the treatment of dangerous diseases like tumors and cancers. Because they are synthetic drugs, these drugs mimic the natural metabolite. So they will get bound to the receptor in the cells. Once they get bound to the receptor, they do not perform the function of the natural metabolite. Instead, they suppress cell growth, division, and proliferation, which can be used in the treatment of certain diseases like cancer. The primary use of antimetabolites is in medicine to treat various conditions, particularly cancer, where they target rapidly dividing cancer cells which are more sensitive to disruptions in their metabolic processes compared to normal healthy cells. The idea is to selectively damage cancer cells while minimizing harm to normal cells. So, normal cells are less affected by antimetabolite disruption as compared to cancer cell disruption. So, selectively, for causing more damage to cancerous cells, antimetabolite drugs are used. Some common types of antimetabolites and their mechanism of action include folate analogs. Example, methotrexate. These drugs interfere with the synthesis of DNA, RNA, and proteins by inhibiting the action of dihydrofolate reductase, an enzyme required for the production of tetrahydrofolate, an essential precursor for nucleotide synthesis. So, the precursor is inhibited. Without sufficient nucleotides, cancer cells cannot replicate their DNA and divide, leading to cell death. So, that is one of the applications of antimetabolites. Folate analogs. So, by inhibiting the action of dihydrofolate reductase, cancerous cells are not allowed to replicate. That leads to their death as well. Pyrimidine analogs. These compounds are incorporated into RNA and DNA during replication and transcription, respectively. Once incorporated, they interfere with the normal function of these molecules, ultimately inhibiting DNA synthesis and cell division. As we can understand, these antimetabolites stop cell division. They do not allow DNA replication, so they stop the cell division. Purine analogs. We know that DNA contains purine and pyrimidine bases. So, pyrimidine analogs, example 5-fluorouracil. Purine analogs are mercaptopurine, thioguanine, etc. These drugs mimic purine nucleotides and can be incorporated into DNA and RNA during replication and transcription. Once integrated, they disrupt the structure and function of nucleic acids, leading to cell cycle arrest and apoptosis, programmed cell death. So, you program the death of the cell by incorporating the purine nucleotides in the DNA and RNA during replication and transcription. Nucleotide analogs, Cytara, cytarabine. These drugs are structurally similar to natural nucleotides and can be incorporated into DNA, resulting in chain termination during DNA synthesis. This effectively halts DNA replication and cell division. So, DNA replication is arrested because premature chain termination takes place. So enough quantity of or enough extent of DNA replication does not occur. So this effectively halts the cell division. Then antiviral nucleotide analogs. Example, ejidothiamidine, AZT. While primarily developed as antiviral drugs, some nucleotide analogs have also been used in cancer treatment. AZT, for instance, inhibits reverse transcriptase in retroviruses such as HIV, and it has been tested for its effects on cancerous cells as well. So nucleotide analogs, they find application both as antiviral drugs and also for arresting cell division in cancerous cells. These are some of the antimetabolites: 5-fluorouracil, mercaptopurine, methotrexate, cytarabine. Antimetabolite uses. A word of caution. If antimetabolites can be highly effective in targeting rapidly dividing cells like cancer cells, they can also affect healthy cells that have a high rate of division, such as cells in the bone marrow and gastrointestinal tract. So, we need to be cautious because they not only affect the cancerous cells but even healthy cells which undergo rapid division are also affected. This can lead to side effects like bone marrow suppression, gastrointestinal disturbances, and other systemic issues. Medical professionals carefully consider the balance between the potential benefits of treating cancer and the possible side effects when using antimetabolites or any other cancerous drugs. The choice of treatment depends on the specific type of cancer, its stage, the patient's overall health, and other personalized factors. So, the word of caution is that antimetabolites need to be used carefully because they can also adversely affect fast replicating healthy cells. If the replication in healthy cells is also retarded or suppressed, it can lead to dangerous side effects. In a nutshell, we have learnt that metabolites play a crucial role in biochemical processes and they also find extensive application in the field of medicine. They are the original chemicals that are found in living systems. Primary metabolites are carbohydrates, proteins, lipids, and nucleic acids. They mediate core metabolic processes like growth and reproduction. Secondary metabolites are pigments, flavors, etc., needed for defense and communication with the environment. Many functions of secondary metabolites are not established even today. Antimetabolites are a category of drugs which interfere with normal metabolic processes by mimicking the natural metabolites. Medical professionals consider the balance between the benefits and the possible side effects when using antimetabolites for cancer treatment. Thank you.